Well, thank you for all of those enlightening presentations and highlighting many of the different variables that contribute to individual variability. I also want to thank you for keeping your presentations within the time allotted to allow adequate time for discussion. I will now open this session for questions. Please type them into the chat box and include the name of the speaker to whom you are directing the question, if relevant. Please note that due to the limited time we have, we may not be able to get to all of the questions today. And hold on a second. The first question that we received was from Bert Garza, who's a member of the Food Forum. And he says that Dr. Mathers raised the importance of interindividual variability in thinking about precision nutrition. One of the questions he raised is whether the field's understanding of interindividual variation would be enhanced by framing interindividual variability as a reflection of the sensitivity of explicit biological pathways to bidirectional complex links to genomic, social, psychological, dietary, and other exposome components to phenotypic outcomes of interest, rather than viewing social, psychological, and biology as separate domains. And I'll open that to anyone that would like to answer it.
Yes, well, I'll jump in it, but someone else should come after me. So obviously that tends to be how people study things now, one domain at a time. And I think, that's clearly what we're keen on, trying to study all these things at once, because they're not necessarily linear systems, meaning 1 + 1 doesn't always equal 2. Sometimes these effects are synergistic. So we think it's really, really important to study them all together so that you can better understand that. And to be honest, there are plenty of modeling algorithms out there that can take this into account. So you can actually account for nonlinear systems to be able to see, what the effects of certain things are, both by themselves and in combination. So I think that's why it's really, really important to study them all together. So I think you've raised a really important point.
Would anyone like? Go ahead.
I would love to ask Dr. Snyder, my perspective being kind of psychology and triggers of appetite, for example, is that some information that you're also kind of getting within your kind of smartphone app, or have you thought about integrating something like that?
We have, but we're not nearly as sophisticated as you. In fact, I loved your talk. I really want to get your questionnaires and bring them in. We do take food surveys, and we also do try and get people to do chronometer. We did MyFitnessPal, but it didn't seem to work quite as well for us. So anyway, we are logging things, and we are taking, trying to get people's perceived stress levels with certain validated questionnaires. But we don't go through, satiety and some of the things you were doing. So I think that would be really fascinating to incorporate. So we're not there at the level you are for the behavioral side, which I think would be incredible to include.
It would be very interesting to, because the kind of mood and anxiety and stress can be triggers, but obviously that is something we've done, work using EMA, and it's very burdensome for people to be kind of reporting on how they feel all the time, but maybe that is something you could use modeling to do to kind of infer that. Maybe there's some interesting work to do around that as well.
We have, through our modeling, we can actually correlate certain metabolites and obviously stress hormones with, and cortisol in particular, with people's stress levels and things like that. So we've done a limited amount of that. And also we have this lifetime stress survey from George Slavich, which also, believe it or not, has a fingerprint on these molecular profiles. It's pretty cool.
And I could just add a little bit to that. I think one of the challenges here is in terms of measuring all of these non-biological factors. The biological factors are, by and large, relatively easy to measure. We have good tools, they're relatively cheap, and they're robust. But the other areas, much more difficult to measure. And the other challenge is knowing what to do with information, because there are few good theories which link these other measurements with diet and with long-term health outcomes. So there's a whole area of research there that I think this will stimulate.
Yeah, but really, I was going to say the same thing as John. The uncertainty really, it differs quite. The type of data that Susan is generating is so valuable, but it's also hard to precisely the same way as we can measure a molecule pretty well, put our minds to it. So, and then as those models get bigger and bigger, our ability to action on that also gets diluted because you can say something broad about a large patient, but as Michael even just showed, everyone's response to fiber really changes. But it's really an exciting time for measurements as we see this come together and our precision gets better.
Okay, the next question is for Abby, and I'll let anyone else jump in afterwards, but is what do you think accounts for the person-to-person variability in the effects of specific foods on gut microbial species?
I think there are a couple possibilities here. The first is that our dietary assessment methodologies often miss information about food preparation, cooking practices, the addition of herbs and spices that could be quite impactful. But one of the things that I really think is going on is that microbes are not breaking down these products alone. They are cross-feeding and interacting with one another, and interacting across kingdoms, right? So fungal and bacterial cross-feeding networks that ultimately, I think that likely accounts for a large amount of the variability, especially when you consider the data that Michael showing some of the variability that you see even when you're providing isolated fiber supplements that are controlling for that difference in preparation, for example.
Does anyone else want to add to that?
Yes, we should think about the microbiome as an ecosystem, very complex ecosystem with all kinds of organisms at all levels, from the guys who are doing the hard work, breaking down the dietary fiber and doing that kind of work, and all the predators, the viruses and so on, that are feeding off those hard-working guys. And trying to understand that complexity and trying to understand how that is influenced by the individual components in food is a major task. And I think it will be a very exciting area as this work develops.
Yeah, and maybe to add to that too, you may know that you have more immune cells in your gut than anywhere else in your body. And so the microbiome, gut, food access, and then metabolome, I think it integrates everything the speakers are putting out there today, is so critical. And it's very easy to perturb your immune system by a variety of means, including your diet. And so I think that's an important part of this whole axis too.
The next question was for Connie, and should we change nutrition labels to include amounts only and not percent of daily intake?
Okay. Actually, I'm not at all sure, really. And I feel that the principal issue is that we somehow have to change the message. And the message needs to be that we have to look at genetic populations. But because of that, I don't know what the best way to do it is. And even in terms of labels, here I'm talking about these different ethnic groups in America have so many problems with different, with people and their attitudes and so forth and so on. I'm thinking, oh no, that wouldn't be good to start listing different, ancestral populations. But there are only a couple of cases where one ancestral population has a major genomic difference relating to one particular element. And I think messaging and education might be a much better way of getting at it. And it's something that I think we're very hesitant about, because we are a multi-ethnic society trying to, keep it together and so forth. And it's a mess. It's an issue of how we do that. But my primary point here is that up until this point, a lot of times when we feel as though we're aggregating data in a universal way, it turns out that if you have 1 million participants who are of European ancestry and 2,000 who are of West African ancestry, you think, oh, well, I'm diversifying. But my point is that diversifying doesn't work very well. We have to stratify if we really want to get accurate data.
And I see other comments coming in regard to this. So does anyone else on the panel want to add additional comments?
I think a nice contrasting point is between Michael's talk and Connie's in a way, because it asked the question, what might be better? What's easier, to just measure sodium levels in everyone and try to get a sense of those people, or to just use people's genotypes, which actually is also part of Michael's technology too. So, and judging whichever is going to be functionally best in identifying those populations and educating them could be good, but it's very different to say African-American descent versus your genetic sodium mutation.
Yeah, let me just say something. I think you're absolutely right. And maybe part of the issue is the fact that we actually do not have data on populations other than people of European ancestry. And so we have this fantastic tool that can do all these things. But the reality is that if we don't stratify populations, then everything is going to end up being the majority, essentially. And so I think we need to appreciate who we are as Americans, because most countries are essentially a particular genetic population, and they've been able to create boundaries, or maybe two genetic populations. Here, America has done something that no other country has done. And so we've reached a point where at a scientific level, it's like, oh, wow, this is more complicated than we thought. And it's more complicated because it's not about races, but it's about different ways of configuring populations related to their health disparities, such as, African American, 75% now. I wonder what that's all about. And so I just feel as though we need to figure out ways to stratify populations and stop thinking in terms of, we'll just put everybody together. But the reality is that we're not going to find out anything about anybody except maybe the majority.
Yeah, totally agree. But picking up on Christian's point, I think both have value. I think the genetic tests, obviously, they're great because they give you pointers, they give you a heads up. And they also probably can give you clues about what your response might be as you try certain treatments, what directions you might go. So I think the measurements as well as the predictions always turn out to be valuable in the end.
Yeah, I agree.
Yeah.
So that we received several questions regarding the affordability of the technologies Dr. Snyder presented on. How do we ensure that people have access to, say, LDL measurements and other blood measures potentially from a finger stick, much like blood glucose? Such technology would bring precision nutrition to the populace. So I'll open that up to anyone that would like to answer.
Well, I could start. I mean, what we presented is ridiculously expensive. Even the version that you go get at QBio costs $3,500 a visit. But it does show, I would argue it shows utility. And then the hope will be, because it's all preventative. And if you can prevent heart attacks, you save a fortune. And I think in the cardiovascular disease space, you can actually make arguments that even when fairly expensive, you will save money if you do roll this out to an at-risk group. But then at the other end of things, what you're saying, just the basic measurements of LDL and things like that, there's a lot of groups working on these home tests for this. And I hope it does get out there for like $10 a test. As you can see, you can now get a pretty cheap COVID test. There's no reason not to get a cheap LDL, glucose, and other very simple biochemical measurements for the same price. So I think it can get out to the world. And I'll make one last plug for wearables. Actually, even a cheap wearable device you could put on everyone on the planet. The system we built could actually scale. You may know that 60% of the planet has a smartphone right now, even in rural parts of the world. And pair that with a wearable device, and you've got a health monitor for 60% of the planet, and a $60 device could do everything I showed you today. So I actually think some of the stuff can get out to the whole world. And we're trying to do that actually in some of these remote groups and some of these underrepresented groups, underserved groups.
Okay, I have a question for Susan. What comes first? The people with the behavior seek out energy-dense, highly palatable foods, or does the availability of energy-dense foods drive the eating behavior? You're on mute, Susan.
Such a great question. The answer is definitely both. But I'm trying to think of the kind of streams of evidence that would kind of you can bring to bear. I mean, obviously, we do see, I didn't talk about evidence for genetics of food preferences. So we do see that they also show a genetic component. So I talked about genetic influence on appetite. We do also see genetic influence on food preferences, which seems to be a bit stronger for fruit and vegetables and protein foods than it does for snack foods. But those seem to show a much more environmental influence. So I think that's kind of relevant here. So I think there is some kind of a genetic aspect. But of course, we can also think of environment as the way that kind of our genetics express. If we all lived in a kind of environment where there were only healthy foods available, then we would all be kind of eating healthily. So I think it's obviously kind of genetics and environment are very important. So we kind of need the environment to kind of express the kind of the genetic propensity perhaps for seeking out highly energy dense foods. Of course, another thing to point out is that we're all genetically predisposed to eat high energy dense foods. It makes sense to kind of seek more calories. So we're talking about those differences between individuals, but there are obviously these broader effects. There's been some evidence in animals, so from animal research where we know that certain animals are predisposed to gain weight on a high-fat diet, that they do actually show some kind of irregularities in food-seeking behavior even before the diet has had its effect on adiposity. So that's kind of some evidence to support that there may be pre-existing differences which drive people in a certain way. That said, there's also lots of evidence that overconsumption of high energy dense foods might actually downregulate dopamine receptors in the brain, and individuals may actually become less responsive to those foods. So they may actually experience kind of less reward, which actually may kind of seek them to promote seeking of those foods. So we also know it's definitely happening in the other direction. And I think that's just kind of a growing area of research, which I could talk about all day. So I'll stop now.
The next question is whether anyone would like to comment on the utility of early detection of disease via genomic markers with respect to overdiagnosis, i.e. disease that failed to progress to pathology. A number of people have also asked about the chance of false positives and whether this would have impacts on patients' well-being.
I can start with that one because we do a lot of this where we actually sequence genomes of healthy people and make predictions. And everybody worried when we were first doing this. In fact, we were told well, this is such a bad idea. It's going to cost millions of dollars in follow-up tests and lead to way too much overdiagnosis. And in the end, we wound up, even in the most aggressive form, leading to about 3 follow-up tests for about $700 on average. So it wasn't that expensive after all. But when you balance it against things like, finding some of the BRCA mutation, who didn't know it? And by the way, BRCA mutations only lead to breast cancer or ovarian cancer in 50% of women. But we obviously think that's really, really important, but you would argue that as a 50% false positive rate. Yeah, I think all of us would agree it's really, really important if someone has to find out if they have a BRCA mutation, get screened for it. So you really have to think about these numbers in terms of what they really mean in risk terms. And so for the people we've done, they've actually appreciated the ones I mentioned. They all 12 did not have necessarily disease. Some did, but some did not. And cases that did not, they'll just get screened for this when they go in for their routine exams. They'll now drill in a little more in those areas. So I would argue that it's just a screening tool, no different from LDL or other ways. It just gives you another mechanism, biomarker, if you will, for what you're at risk for. And I think that's the way to view these things.
Michael, if I could just follow up with another question related to that. Was there any evidence that the people who were taking part in the continuous monitoring had more anxiety than others? I mean, did it create that kind of unintended consequence?
Yeah, well, we did do a survey at the 3-year, and we've done it again recently. Now, I should point out, you could argue we've selected for an eager beaver crowd. So they loved it. They all agreed, in fact, that we asked them if they'd be willing to make their data completely open, including their genome sequence, and nearly all have. So they're clearly a very motivated, go get them crowd. And so, of course, not surprisingly, 95% said they all got some benefit from the study and how much they liked it. And we didn't see a single comment. We specifically asked for it. The sort of like, did you get increased anxiety or anything like this? It certainly wasn't from our study. It could be from those stress surveys, other things going on in their lives, but not necessarily our study.
So the next question is for Christian. The person said that they loved being able to dissect a disease via metabolomics. Can this be readily scalable to all major diseases?
I think it will be challenging, and because of how much effort has been put over the years. And this also reminded that the MacTel project was made possible by one family who really wanted to combat this over the years with extraordinary wealth and the ability to go in different directions in the clinical. But that said, by studying the molecular mechanisms of this single disease, we can learn about those different types of diabetes, the precision diabetes that Michael was describing. So that's, I think, our best chance, because it's hard to go after all diseases in this way. It also highlights the point that Connie has made, in that the data set, the populations that we generate our data sets from, need to be representative of the overall population, or else we're going to be just studying pockets.
Okay, the next question is for Abby. Can we use microbiome data to give insight into dietary intake when dietary intake was not assessed, or since dietary intake is difficult to capture accurately in large cohorts?
I love this question. This is something I've thought a lot about. It's a big limitation in all of our studies, right? Collecting dietary data is expensive. It's difficult. It's fraught with error. People have a hard time remembering what they ate. And most of our methods rely on recall in one way, shape, or form. So theoretically, I think in the future we might be able to. So Hannah Holscher at the University of Illinois is doing some amazing controlled trials where she's collecting information of how the microbiome responds to single foods, and is starting to get a big enough data set with the same methods used across studies that they're able to now try and predict whether or not a person ate almonds or broccoli. There's also work out of Lawrence David's lab where they have taken the study where they put people on a plant diet versus an animal diet and done metabarcoding. So this is kind of stealing from the world of trying to understand animal diets, where can you, instead of looking at the 16S barcode that we often use for microbiome, can you do metabarcoding for another DNA region that is specific to plants, and try and get information about the diversity of the plants that the person is consuming. There are challenges, because if you know anything about plant genomes do not behave nicely. They are filled with all sorts of things that break these assumptions. So maybe in the future, we'll get to the point where we have a series of barcodes that we could use on a sample, or if we know enough information about the person's species microbiomes, we might be able to predict maybe their habitual diet, things that they usually eat. Because we find agreement between diet and microbiome without that Procrustes analysis, that suggests that we can predict one from the other. And we did try to predict tomorrow's microbiome from today's microbiome, incorporating to yesterday's diet. And we find that that prediction actually works relatively well and improves the prediction over just saying, well, tomorrow's microbiome will just be today's microbiome. So we know that we can do it in that direction. Whether or not you can do it the other way, can you predict yesterday's diet from tomorrow's microbiome or today's microbiome? I think that's a harder problem because there's just so much that a person could pick to eat. So my answer is maybe, hopefully, yes.
Thank you. So this
Maybe
question,
at that point, it would be great to integrate what Susan's doing with what you're doing, Abby. So put that be, what behaviorals get stimulated by, which microbes.
I love it. I think we need another ohm, a behavior ohm, so we can add to genome, metabolome, microbiome, and behavior ohm for sure.
In the short term, while Abby's sorting out the complexities of the gut microbiome, it might be that the metabolome will be a quicker route to finding out what we eat, because it's so much directly related to the individual food. So I suspect that metabolomics on blood or urine or even saliva might be a way forward.
so that actually sets it up perfectly for the next question. How well understood are the role of comprehensive metabolome and metabolite differences in interindividual variability as it relates to microbiome mediation or host-derived variability? What evidence gaps or research priorities would be most critical to advance this understanding?
So I think there is a fair bit of variance if you pick any particular metabolite, and that's why we tried to get hyper-focused on a particular pathway and then branch out from there. And then why we've also tried to move forward and look at more of the dynamics. Think about a serine tolerance test, analogous to a glucose tolerance test, might give you a better readout because by and large, given the fluctuations between the feeding and fasting states and the circadian rhythms, even amino acid levels fluctuate to a good degree, and serine and glycine levels fluctuate differently as well. But as we get deeper, we can back out into these more surface level measurements. And the other challenges look really mostly a readout of liver and kidney function when you're starting to think about the nervous system, or the central nervous system. It's hard to access those metabolites.
Yeah, maybe to pick up on that point, I think the links, for example, between autism and metabolites and the microbiome, the work of Sarkis, is really fascinating. And I think you're seeing more and more of this come out. So to the questioner's point, I don't think we know very much at all about this area. There's a ton to do. And I think it really needs to be done on humans, to be honest. There's a lot done on mouse, but it's just hard to translate from the mouse to humans. So I think these dietary studies and the purpose of this workshop is really, really important, because I think we do need to make these relationships between food and metabolite and microbiome. They're all connected. And again, I think we need to bring the immunome in on this, because it's so tied into the microbiome and then therefore your metabolome. It's a big deal.
Another critical aspect of this, which links the human genome with the metabolome, is the phase 2 enzyme system, which is involved in converting the metabolites into things that are easily soluble and excreted in urine. And that's highly polymorphic. So understanding all of that genetic variation will help us to understand the metabolomic variation.
I'll leave one other thing in there that we've left out, which is the epigenome. There's a lot of studies, some beautiful studies suggesting, your DNA methylome is affected by nutrition and goes transgenerational. And I think that's going to be huge. I mean, again, I'm type 2 diabetic, and my methylome changed the 2 times my glucose control went off. So, and the very understudied area. So, and it turns out the methylated regions in my DNA that went off turned out to be upstream of my metabolic genes. So I think this idea that, again, your epigenetics can be influenced by your nutrition and affect your chronic phenotype. It's already known for aging, right? That your methylation changes with age. Obviously, your metabolism changes with age. And I think understanding this whole relationship is really, really important. That's an area that's pretty much untouched, I would argue, the food epigenome relationship.
You kind of got us going into the next question, is that we've received several questions regarding ageotypes. What are the main biological mechanisms possibly explaining why there are so many ageotypes? different individuals' aging paths? And regarding classification, what would it take for these classes to be applied for prediction of an individual's susceptibility to age-related disease?
Well, that one's probably for me. We think they're all tied, obviously. So taking care of your immune system, one of the ageotypes is an immunoaging phenotype. And so obviously keeping a good, healthy immune system is a big deal, probably for all chronic diseases, including cancer. And so I guess, we can measure it. This is step one. That's what we're trying to do now, measure these different things. Step two is, and trying to understand, I didn't get into this, but there's actually clinical markers associated with each of these ageotypes. So for example, obviously, glucose and hemoglobin A1C is an easy one with the metabolic. It's more than that though, but that's one of the markers associated that you can follow. And we can follow this at an individual level. You'll see the population as a whole all goes up for their hemoglobin A1C with age. That's no surprise. But what's kind of cool is you'll see some people go way up. Other people actually reverse that. What did they do to reverse that? Well, it's obvious. They did exercise and things like that. But then there's another one I don't understand. It's creatinine, which is not a marker of kidney function. So that's for the kidney ageotype. And that's a case, again, the population as a whole went up, but 10 individuals went down. 8 of the 10 were on statins. And I don't think they lost just muscle mass. It could be, but it wasn't obvious. So something's going on with statins for improving their creatinine and presumably kidney function. That's a hypothesis, needs to be followed up. But this is the kind of stuff you can learn from these observational studies, but then they also lead to interventions. So if you're a kidney ager, maybe you should drink more water and see if you can reverse your kidney ageotype, right? And liver folks, maybe you shouldn't drink quite so much, although most studies say moderate drinkers live longer, but binge drinkers not so much. So anyway, the point is you can modify your behavior. Maybe if you're an immunoager, you have more garlic or turmeric or things like that. So that fits nicely into the whole diet scheme of things. So anyway, I think this is all, we can follow how people are aging. I will point out that the cardio ager I presented, turns out we learned after the fact was stage 2 hypertensive. So it all kind of fits that phenotypes can fit with these ageotypes we're seeing, again, which it can relate to actionable information. Kind of a long answer for a question.
Actually, if I can make another point, I think, at least from my own research, what I found over and over and over again is that people digest most efficiently the staple foods from their ancestral diet. And what it means is that when immigrants are brought into a new locale, such as immigrants living in the United States and so forth, it's not really all that surprising that populations whose staple food were sorghum and millet, or whose staple food was rice or whatever, that what appears to happen is that the digestive mechanism ages faster if they're eating all these foods that they do not digest as efficiently as what their microbiome and what their genetic variants have sort of evolved to do efficiently.
So the next question I kind of want each person to try and address or think about, but given the limitations of measuring mean response, when do you foresee the current paradigm of clinical trial design change in a sophisticated manner to allow for personalized response to be measurable and meaningful to the conclusion of efficacy?
Well, perhaps I should start since I did the first presentation. I think it's already beginning to change a little bit with precision medicine, because we begin to think about the drugs that will be appropriate for a given individual. The difficulty is that the clinical trial design has such a grip on the research community that it's hard to see how quickly people will be willing to give up what has proven to be a very, very effective trial design to address the new needs. But I think it's going to happen. It's unlikely to be, in my guess, something that'll be in the next few years, but 10 years out, I think it'll be more common. And N-of-1 studies are already happening, and I think we'll see many more of those N-of-1 studies. The difficulty is linking those to claims for any substance that's linked to the outcomes of the trial.
Yeah, I agree completely. I'll just add one comment. I think we need to think more about this concept of responders and non-responders, to John's point, which we don't now. We always look for the final average, just population average. And it's clear that, you know, even drugs that have been pulled from the market can be incredibly beneficial to some people, but they get pulled because they have adverse effects on some. So I think we've really got to pin down the responders and non-responders. I think in the nutrition field, it's probably more critical than any field for that. So we can see this is good for these people, and they're the ones who should take it and benefit. And these others, if they don't benefit, well, that's okay. They don't have to take that. But I think that's the right mentality. And then we need the right trial design and the acceptance going in, because you have to define your endpoints going into a trial, and then they're not accepted if you haven't defined them properly. So we've really got to embrace this new idea that, again, we are personalized in the way we will respond. Just picking up on John's point.
I think adding to that, there's a lot that we can add into our current clinical trial designs by increasing the density of sampling and the number of time points that we're collecting data. So rather than doing a crossover trial where by classical design, you do one at baseline, one at the end of the first arm, one at the end of the second arm, we can add density to our sampling time point to allow us to look at what's happening at that individual level, reducing some of the error from the repeated measures by being able to look at, at time points, what's happening when we get 3 samples for that person, followed by what happened when we took the intervention away. Did those next 3 samples look different? So I really think that increasing density of sampling is going to help. Partly, though, we're limited in this responder, non-responder thing by defining what is a responder. Like I've got data that I'm sitting on where I've got responders and non-responders, and I'm struggling with the definition of how do I call a person's response a response versus versus is that just normal variation and error? So coming up with statistical methods that we can apply that incorporate these longitudinal designs and allow us to detect those actual variations that are meaningful and important. And that you could just define ahead of time, like I'm going to call a responder a change of this much in this thing. Then I think we're getting somewhere.
I would just like to chip in quickly from the behavioral perspective. So this is something we think about in terms of, say, what might be psychological predictors of outcomes in weight loss treatment. And there are some moves to at least try and get some consistency in measures across the field. So I talked about the variety of measures out there. So I don't know how well they'll be adopted, but I can definitely see we might need something ideally where we would combine data and be able to look at least some consistent measures kind of across a bunch of different trials to try and pull out some predictors that way.
So the next question is, again, to everyone, is the current precision nutrition industry relies on early adopters and vulnerable individuals eager to understand how to eat to be healthy and address symptoms or potential health concerns. However, the value that is provided to paying customers is often low due to our limited understanding at the moment. The private industry is well positioned to innovate in this field, but the way emerging applications are marketed is often ahead of the science. What are the practical and ethical considerations that we should keep in mind when recruiting volunteers or paying customers to develop this evidence foundation for the precision nutrition of the future?
I think there's a really important point that we often don't spend enough time on. We are developing technology on and for the worried well, and are potentially leaving a lot of people out of this conversation. And it needs to be front and center. how do you design experiments and studies and technology for everyone, not just for rich white people? We just need to address it front on and talk about it and bring people to the table so that we have more representation when we're having these conversations. Otherwise, it's totally insular and we're not going to get anywhere.
I completely agree. I think what you're saying is so very valid. And one point that I want to inject is that over the course of the last several decades, there's been this, I guess, sort of new field on racial and ethnic health disparities and the gap between various minorities and the majority. And I feel that that kind of research is extraordinarily important because every group is not going to have the same risk for every disease. And the point is to find patterns, because even when we're looking at the individual level, we're sort of looking for patterns. But if in fact it turns out that it's not just going from the universal Homo sapiens to the individual, but it's going through genetic populations, then I think it becomes more and more important for us to just take these kinds of issues into consideration.
Yeah, I 100% agree. I think this has to come from government funding, because the reality is the private sector, it's going to be the rich people who will pay for it. So you actually have to run it as a research, in our case, NIH or USDA funded projects or agriculture. Hence this meeting.
Consequences of inequity affect all of us, affects the whole society. And I think whole society needs to address this issue and be willing to invest in the kind of systems which will enable the people who need to benefit from these systems from being able to access them. So access to personalized or precision nutrition needs to be something which is supported from society, rather than expecting the individual necessarily to pay up front, because the people who need it most probably won't be able to afford to do it.
So we need from the last two questions, we need more measurements, more data, and better, more representative cohorts. But the other thing, oftentimes the patients that can be helped the most or aren't the ones that are going to be getting involved early on. They're going to be already the well-nutritioned, healthiest folks. So it's actually works against the system.
Okay, so we're down to the last 4 minutes. So I'd like to just give each of you the opportunity to make one last final thought that you want people to leave with, thinking about the end of today's presentations. So I'll start with Michael.
Oh, I think this is a very understudied area, and it is the future. I really believe food is medicine. I think we all are personalized. Imagine if every person on the planet could log what they're doing and get some sort of metabolite profile, microbiome to go with it. The information would be so rich that we could really treat people incredibly well at an individual level just through diet. I'm very Pollyannish, by the way, so probably tell.
Connie?
I just want to reinforce the statement that we don't want to forget about the fact that we're dealing with human beings, but we're also dealing with genetic populations who have different sets of variants and microbiomes and so forth and so on. And so we just need to keep that awareness or to inject that awareness into the kinds of research we're doing so that we're not misunderstanding a particular disease because we assume everybody is affected in the same way.
Yep. Abby?
A lot of pressure to come up with something good. I think we're on the cusp of something amazing. We've got a lot of opportunity here, and it's going to be really important that we take our time and do it well and not rush to conclusions too early based on like early data results. And yeah, I'm excited to see where we're headed.
Christian?
Yeah, I agree. I'm excited in the same way. I'm particularly excited, somewhat biased, about molecular detail that we're able to get down to now. Because at its core, the human machine is a pretty complex machine with different genotypes and different blueprints. But when it comes down to it, health issues boil down to the molecular level.
John?
Yeah, this is a really exciting area, and I think this will drive innovation into a whole range of areas of science, not just nutrition, but related sciences. But I think we need to keep an eye on the big picture and ask all the time, how is this improving public health? And what are we doing that will address the inequities in public health?
And Susan?
So as the psychologist in the panel, I'll chip in and say that I think it'll be really important to evaluate how people are responding to this, the types of interventions that we're giving and receiving this information, because we know it's not always how we might expect. And I just think that's something that we'll need to do further research on as this exciting field develops.
Well, great. Thank you very much. That was a wonderful discussion. And I really want to thank the audience for their thoughtful questions. And an especially big thank you to the wonderful speakers for their enlightening presentations and remarks. We will now be adjourning the workshop for the day, but please join us tomorrow, August 11th at 10 a.m. for the second session on innovative methodologies and technologies in precision and personalized nutrition. Thank you again and enjoy the rest of your day.